Hey everybody, so we are going to continue on talking about photosynthesis, and we are going to take a look at the next set of reactions. So last time we talked about the light-dependent reactions. Now we're going to look at the light-independent reactions. And so this is how plants are actually making their food, or the carbohydrates. And so this part doesn't require light directly to run. However, the energy from light that's captured during the light-dependent reactions, that is going to be sent over for use in the light-independent reactions. This is also going to be known as the Kelvin cycle. That's a picture of Melvin Kelvin, who this cycle is named after, or it's also called C3 photosynthesis, because at the very end you're actually producing these 3-carbon sugars that can then be used to make larger sugars like glucose or even starches. So the light independent reactions is going to happen in the stroma of the chloroplast, and the function is to use ATP and NADPH to build sugars from carbon dioxide. So we know that carbon dioxide is an overall reactant in this process, and so the carbon dioxide is going to go directly into the stroma for the light independent reactions, so it can get converted into sugar. We want to talk really quickly about something called ribisco, an enzyme. So the full name is ribulose biphosphate carboxylase, and this is an enzyme that actually adds carbon dioxide to an acceptor molecule. Some people say this is the most important enzyme on Earth. And the reason for that is because it's what's pulling this carbon dioxide into the process of the light-dependent reactions, or the Kelvin cycle. And then it's because that carbon dioxide is pulled in, you are able to make sugars and kind of, provide energy for everything on Earth. So that's how you are providing the energy and the food for every living thing, is because plants are able to make their own sugars, and they're able to do so because this carbon dioxide is pulled into the Kelvin cycle. And so, yes, ribisco is very important, and we're going to see where it is sort of playing its role in a Kelvin cycle in a minute here. So here are the requirements and the products for the Kelvin cycle. So we're going to look at 6 carbon dioxides. Those 6 carbon dioxides are going to get changed into a molecule of glucose. So since you've got 6 carbon dioxides and 6 actual carbons, then you can produce a glucose, which is made of 6 carbons. You need 18 total ATP, and so after this you will have made 18 ADP and phosphates, and you need 12 NADPHs, which means you will use those 12 and create 12 NADP pluses. So here is sort of a breakdown of the Kelvin cycle. So you've got your 3 carbon dioxide. So keep in mind, before I said 6, and here I'm showing you 3. And so for 1 molecule of glucose, you're going to have to go through this cycle twice. So you've got your 3 carbon dioxides up here. They're being pulled into this cycle by ribisco. So this is the first step. We call this phase carbon fixation. And so the carbon is being fixed or pulled into the Kelvin cycle. And that is happening with ribisco right here. Now, what I want you guys to look at is, before this carbon dioxide is actually pulled in, you are starting this phase with this RUBP, ribulose biphosphate. Okay, so you have 3 of these RUBP molecules. Okay, so it's biphosphate. You've got a phosphate group on one end and a phosphate group on the other end. So there's your 2 phosphates. You have 3 of them, and they are 5 carbons each. So 1, 2, 3, 4, 5. Okay, so total you are looking at 15 carbons, because you have 3 of these molecules and each of them has 5 carbons. Okay, then ribisco is going to fix 3 carbon dioxides. So you're adding 3 carbons to these 15 carbons. So you're going to end up with 18 total carbons, moving on to this next step. So you've got these 18 carbons. There's a lot of enzymes at work here. You're going to have lots of intermediates formed. You guys are not being held responsible for all of that. So what you do need to know, though, is that you are going to end up converting this into 6 3-carbon molecules. So now you have 1, 2, 3, okay, but you still have your 18 because you have 6 of these 3-carbon molecules. 6 times 3 is 18. So we started with 15, we pulled in 3 carbon dioxides, we changed it, kind of rearranged everything, so but we're still looking at 18 carbons, okay? Notice here we're looking at 1 phosphate group per each of these molecules. You're going to bring in 6 ATP, so now you have 6 3-carbon molecules, but now each carbon molecule has 2 phosphate groups, okay? Now we're into phase 2, which is the reduction phase. When we talked about cellular respiration, we were oxidizing that sugar, right? So we were breaking it apart to release energy. Here, this is us making sugar, and so this is the reduction phase. So we are going to use up 6 NADPHs. We are going to also release 6 phosphates, and so we're rearranging this molecule again, and we've got 6 G3P molecules. So again, we've got our 3 carbons, and we've got 6 of them, so we're still at 18 carbons, and now only 1 phosphate group per molecule. Now, for every turn of Calvin cycle, one of these 6 molecules is going to be released. So that's what's happening here. So here we are releasing 1 G3P, and so we've got our 3 carbons leaving. So remember, we started with 15, we added 3 carbon dioxides to get to 18. Now we're releasing 3, so we're back down to 15 carbons in this cycle because we released one of them, and this is a sugar, okay? So this is 1 sugar with 3 carbons being released. All right, now we are going to move on to the regeneration phase. And so the regeneration phase is going to come in. Now we've got 5 3-carbon molecules, so we're at our 15 carbons. We're going to add some ATP, we're going to add 3 ATP, and as we rearrange these carbons in these molecules, we are going to regenerate, so this is our regeneration phase, we are going to regenerate that ribulose biphosphate. So now, still at 15 carbons, so 1, 2, 3, 4, 5, but we have 3 of those molecules. Remember, we added 3 more phosphates, so now you've got 2 phosphates on either end, or 1 phosphate on either end, so 2 phosphates total per molecule, and you are ready to go through this cycle one more time. Okay, so with that one turn, we created 1 3-carbon sugar. Okay, so in order to create a 6-carbon sugar, we would go through this again, release another G3P, and then those 2 G3Ps can combine to make other sugars and starches that are needed for or needed by the cell. Okay, so here is sort of everything together, showing you where all this stuff takes place. So you're going to have your light dependent reactions. Water and light are going to come into this reaction. This is taking place in the thylakoid membrane. So you're going to have the light activate photosystem 2. You're going to send those high energy electrons through the electron transport chain. They're going to get re-energized in photosystem 1. With photosystem 1, you're going to generate ATP and NADPH, which will travel over to the Calvin cycle, and you're also going to release oxygen. Okay. Now, when we look at the light independent reactions or the Calvin cycle, we have carbon dioxide coming in. Okay, it's going to go through that Calvin cycle. It's being pulled in by Rubisco. Things are going to be changed around. The molecules are going to be kind of converted to those 3-carbon sugars. Here we have G3P, which is going to be released, and then you can have multiple of those G3P molecules come together to make larger sugars, or they could make different starches in order to store energy more long term. Okay, so now this cycle, this Calvin cycle, is using the NADPH and the ATP that was produced in the light-dependent reactions. Okay, it's going to rearrange these molecules some more so you can regenerate the RUBP. Once the ATP and NADPH are used up, they will then cycle back to the light-dependent reactions, pick up more energy, and then carry that energy again over to the Calvin cycle. Okay, so you've got 2 cycles happening here. When you're looking at it overall, you have the Calvin cycle happening over and over again in order to produce these smaller sugars that can be then put together to make starches, or they can make other sugars by combining. You also have the cycling of the NADP plus and ADP into the light-dependent reactions to pick up energy and electrons. Once they have those energy and electrons, you have ATP and NADPH, and they will cycle back to Calvin cycle to deliver the electrons and the energy. Once they're used up, they will again make that trip back to the thylakoids to pick up more. All right, so hopefully you guys are kind of following along with those two cycles. I do want to start talking really quickly about something called photorespiration. And photorespiration is when Rubisco accepts an oxygen molecule instead of carbon dioxide as the substrate. Now, overall, this does cause a loss of ATP and decreases photosynthesis output by as much as 25%. So we talked about this whole process, and the key to this, we even said Rubisco is one of the most important enzymes on Earth because it can pull in this carbon dioxide. We now know, though, that with photorespiration, there can be an error in that, and you're pulling in oxygen. And oxygen does not have the carbons needed to make this G3P molecule. Okay, and without those carbons, you're not actually adding any carbons, so this cycle is not ever getting up to 18 carbons. You're going to be staying at that 15. And so I want to ask you guys a question. Obviously, this is not something that's beneficial for photosynthesis. It's decreasing the output. It's costing the cell ATP. Scientists think that photorespiration may reflect a time when oxygen was less plentiful and carbon dioxide was more common. And so I want you guys to think about why photorespiration would happen, why Rubisco could possibly pick up oxygen instead of carbon dioxide. Think about what the Earth used to be like, what our atmosphere used to be like, and I want you guys to try and explain why photorespiration could occur, why it's even possible for Rubisco to pick up oxygen instead of carbon dioxide. Send me a quick comment from the website with your answer to this question, and I will see you guys soon. Thanks.